Very good morning, everybody. Today we'll start again from where we left last time. Uh, the topic was polymerase chain reaction. And this particular topic, you should know that although we are trying to understand the introductory aspects of PCR, that is polymerase chain reaction, from the instrumentation point of view, we should know something more about the instrument, and that is what I'm going to cover at the end of this particular topic. But before that, we will first try to know what is the polymerase chain reaction, some detailed aspects about the reactants that are used, how the reaction is set up, okay, and then probably we will cover types of PCR reactions, and then we will go for the instrumentation part. Is it okay? So last time we have seen about the basic introduction about the PCR, polymerase chain reaction. And then I told you that this is an enzymatic reaction which is carried out in vitro. Okay, without the living cells it is carried out. So it is in vitro. But we are using the biochemical reagents. We are using the biocatalyst, that is the DNA polymerase enzyme. Okay. Can you hear me? Somebody's mic is on.
Yes, sir.
That was creating some problem. So we have the template DNA which is to be amplified, and this DNA at a particular high temperature of say about 90 degrees, this DNA is subjected to what is called as denaturation. Denaturation here means that there is something called as the strand separation. Okay, so the two strands of the DNA, they separate from each other, the hydrogen bonds are broken. Okay, and then each template strand will serve as a a template for the synthesis of new DNA molecules. Okay, so then in the next step we lower the temperature to say about 50-55 degrees, and at this temperature when we add the primers are going to stick to the two extremes of the DNA. Okay, that is at the 3' ends of both the strands. And then in the next stage, that is the third stage, we again increase the temperature to about 72 degrees. And at this temperature, the polymerization reaction will take place. So that is because we are adding DNTPs to the reaction mixture. Okay, these DNTPs are going to one by one they will get added to the growing strand, depending upon what bases are present in the template strand. So there is definitely complementary pairing, and therefore the complementary type of DNTPs will be added to the growing strand. Okay, and that is how we get the DNA synthesis. Now these particular cycles are repeated again and again for at least say 30 times, or sometimes maybe 35 times or 40 times, so that the required amount of the DNA is generated, and then the reaction is stopped. Okay, so basically we should understand that whatever reagents are added to the mixture or raw material is added to the mixture, that the quantity of the raw material should be enough so that the 40 cycles of the DNA synthesis will take place, and at the end of the 40 cycles or 35 cycles, we will get enough amount of the DNA. Okay. Then I told you that this reaction can be carried out in two ways. Either we can carry it out manually in water baths. That is what was done in earlier days when the thermal cycler machine or PCR machine was not available. Okay, so people used to carry out these reactions manually. The water baths were set at 3 different temperatures, say 95 degree or 94 degree, 55 degree, and 72 degree. And then the wires containing the reaction mixture were placed in each of these water baths one by one. Okay, and that was done for the required number of cycles, so that the DNA synthesis took place, and an amplified version of the DNA was obtained. Okay, the other way is to carry out the polymerase chain reaction in a specialized machine that is called as the PCR machine or the thermal cycler machine. Okay, about which we are going to see a little later. And then I told you that a large number of DNA copies are made. We get to the tune of something like 10 raised to 9 or 10 raised to 10 copies of target DNA. And there was one small assignment which I gave you. And I told you that if there are, say, 10, how much is that? 10 raised to 21 stars are there in the universe. Okay, so to get that number of the DNA, how many different reactions will you have to carry out? Considering that about 10 raised to 9 copies of DNA are made in one reaction tube. Okay, so how many reaction tubes you have to use so that you will get the number of DNA equal to the number of stars in the universe? How many of you have done that exercise? I think I got only one assignment from one student. About others?
Sir, I was unable to do that.
Okay, but will you be able to do it in the next 1 or 2 days? Yes. If I give you one or
Sir, I cannot understand the
you
problem.
can, what is your question?
Sir, I am not properly understanding. I tried but failed. I cannot understand
Doesn't matter.
actually.
So whatever you have tried, you can just give me that paper. Okay, you can just take a snapshot of your exercise and then give it to me. Okay.
Okay, sir.
So once I get it from everybody, then I'll be able to explain you. Okay. Well, then we were talking about the first reaction component, and that is the DNA polymerase enzyme. Okay. Now this DNA polymerase enzyme is also called as DNA Pol1. This is taken from E. coli. Okay, you know everybody. E. coli is a very popular and very commonly used organism, the model organism, I would say, which has been investigated in a lot of detail. So the enzyme called DNA polymerase from E. coli has been used conventionally for carrying out PCR reactions. Okay. Then subsequently, what the people did was they took out a particular part of that enzyme, okay, not the entire enzyme, okay, and this particular part or fragment of the DNA polymerase was found to be enough to carry out PCR reactions in the test tube, and that particular fragment of the DNA polymerase enzyme is called as the Kleno fragment. Okay, the speciality of this particular part of the polymerase is that it has 5' to 3' polymerase activity. Now, what is 5' to 3' polymerase activity? Let us go back to my earlier slide, this one. Okay, so you see that the primer was attached here at the 3' end of the template. Okay, so the primer has this as the 3' end, and this is the 5' end, right? And then the new synthesis will take place at this 3' end of the primer. Okay, new nucleotide will be added here at this point, right? And therefore this end of the primer will grow. Okay, it will become long, and ultimately the entire strand of the DNA will be made. So this synthesis actually is taking place from the 5' end of the DNA to the 3' end. Okay, now this activity, this polymerase activity is called as 5' to 3' polymerase activity. Is it okay? Please switch off the mic. Somebody's microphone is causing some trouble, some disturbance here. कौन है रे? Meenal, Meenal, can you hear me? Okay, so I think I have muted that microphone, and we will go back to our discussion. Okay, so this particular thing is very important. 5' to 3' polymerase activity is there. That is essential for us. And the other thing is that this particular enzyme can also carry out what is called as the proofreading activity. Okay, what is proofreading activity? It is to check whether the nucleotides which are added in the strand are correct or not. Okay, and if there is any incorrect nucleotide added to the strand, okay, that is not complementary to the template, then that nucleotide is automatically removed by the same enzyme. Okay, so that is called as proofreading activity or repair activity, you can call it. Okay, and that is from the 3' to the 5' end of the DNA. So again, let us go here. 3' to 5' end repair activity or proofreading activity actually means that if a wrong nucleotide is added, okay, if a wrong nucleotide is added somewhere here, yeah, and the growing strand, okay, then the proofreading activity, because of the proofreading activity of the polymerase, that particular wrong nucleotide will be removed by the enzyme. Okay, so that activity is from the 3' end to the 5' end. Now, for example, if by mistake 5 nucleotides are added, which are wrong, okay, then from this end of the DNA strand, that is 3' to the 5' end, one by one, all of those 5 nucleotides, wrong nucleotides, will be removed by the enzyme. And then once again, the polymerase activity will start. Is it okay? So this particular Kleno fragment has both the enzyme activities. It has 2 enzyme activities. In other words, one is the polymerase activity, that is adding the nucleotides one by one. And the other activity is the repair activity or proofreading activity, whereby the wrongly incorporated nucleotides are removed one by one from the 3' end to the 5' end. Have you all followed what I'm saying?
Yes, sir.
So the use of this particular enzyme is that it can therefore repair the mismatches at the ends, and it can also fill the gaps in either strand. So if there are any gaps which are remaining in the strand, and those gaps can also be filled by this particular enzyme. Okay, but the problem with this enzyme is that this enzyme is not a heat resistant enzyme, and therefore at a higher temperature the enzyme gets inactivated or it gets denatured. Okay, now imagine that we have done the DNA melting, okay, or strand separation at 94 degrees. Then in the next step, the primers will be annealed. And then in the third step, the polymerase activity of the DNA polymerase enzyme will happen, isn't it? Now what happens is when the second cycle is there, when the second cycle starts, then again the temperature will go to 94 degrees. At the 94-degree temperature, the whatever DNA strands are synthesized, new strands, they will get separated from each other. Okay, and then there will be primer annealing at the next round, next step. Okay, and then in the third step, the DNA polymerase enzyme is supposed to carry out the DNA polymerization, okay, or synthesis of new strands of DNA. But the problem is, because of that 94-degree temperature which was employed, the DNA polymerase is losing the activity. And therefore, in the subsequent cycles, probably the enzyme is not active, and therefore the DNA polymerization will not take place. Are you following what I'm saying?
Yes, sir.
Yes, sir.
So what happens is, now if you want to carry out the second cycle, then what are we going to do?
We need fresh enzymes.
Yes, yes, we have to add a fresh enzyme again. Just a minute, I'll take a pause for a. Yeah, I'm back. There was some disturbance in the corridor, so I had to go out and tell somebody not to make noise. Okay, so I was here, I was telling about DNA Pol 1 or Kleno fragment taken from the DNA Pol 1 of E. coli. Okay, and I told you that it has 5' to 3' polymerase activity. It also has 3' to 5' exonuclease or the proofreading activity. Okay, exonuclease means what? It cleaves the DNA, it removes the nucleotides from one end of the DNA, not from the middle of the DNA, but from one end of the DNA, that is from the 3' end of the DNA to the 5' end. Okay, so that is called exonuclease activity. And this enzyme can also repair the mismatches, it can fill up the gaps. Okay, but the problem with this enzyme is that it is inactivated at a high temperature. Now we in our subsequent cycles we are going to increase the temperature to 94 degrees, then we are going to reduce it to 55 degree, again increase to 72, again increase to 94 degree, and when we are going to repeat the cycles again and again, that enzyme is going to be inactivated. Okay, and therefore, as I think Shamil told me or Inamul told, I don't remember, he said that we have to add the fresh enzyme again and again for every cycle. And the answer is correct. We have to add a fresh alicot of the enzyme every time for every cycle. And that sometimes becomes very cumbersome. It becomes a difficult kind of a work. Okay. And something must be done about it. And therefore, what the scientists have done is they have successfully isolated some thermophilic organisms. Okay, and from these thermophilic organisms they have obtained or they have purified the enzyme DNA polymerase. Okay, and that DNA polymerase is temperature resistant. Okay, and that can be used at higher temperature without any alteration in the activity of the enzyme. Okay, then another feature about this particular Kleno fragment is that it can amplify about 400 base pair of DNA. Okay, so 400 base pair actually is a small stretch of DNA. It is not a very big amount of DNA. So small, you can say small molecules of DNA can be amplified using this particular enzyme. Is it okay?
Yes. Yes, sir.
Then we move on to another important slide, and that tells us that a particular type of polymerase, which is called as TAQ polymerase, TAQ. This particular enzyme is taken from a thermophilic organism, and that is called as Thermos Aquaticus. Okay, Thermos Aquaticus. This particular organism is actually an archibacterium. I think you might have already come across these words, archibacteria or extremophiles, maybe in your first year of BSc when you studied different types of organisms.
Yes.
Okay. Do you know about it?
Yes.
So this particular organism, Thermos Aquaticus, okay, that yields an enzyme, polymerase enzyme, which is called TAQ polymerase. Okay, this organism has been isolated from hot springs, hot water spring. Okay, and about 0.5 to 2.5 units of this enzyme is added to the reaction mixture, which is say about 50 microliter, and that actually means that we are adding about 2 into 10 raised to 12 to 10 into 10 raised to 12 molecules. That is 10 raised to 13 molecules per reaction. Okay, and the specific activity of this enzyme is 80,000 unit per milligram. Now, I don't think that these numbers are very important from your examination point of view. We are not going to ask you questions about what is the specific activity of a particular polymerase enzyme, but this is only for your information. Why information? Why this is necessary? Because you should know that these are some terms which are associated with enzymes. Okay, specific activity, for example, is a term which is associated with enzyme, and you should know what is the definition of specific activity, because these questions will surely probably will be asked in your competitive exams when you appear for NET, SET, PET or GATE exams in the future. Okay. Now the yield of this enzyme is also very good. It gives a higher number of the sequences which are amplified. Okay. Then this is another important thing. When we anneal the primers, there has to be a certain particular temperature which is required to be set, because normally what happens is at a low temperature or ambient temperature, the primers do not anneal to the particular site in the DNA. That is, there is a non-specific binding of the primers. Okay, and non-specific binding of primers will often lead to erroneous sequences which are being amplified, and the product which you get will not be the desired product. Okay, and therefore, if the temperature is raised to say some temperature, say more than 70 degrees, then the binding of the primer to the particular sequence in the DNA will be very specific. Okay, and because of the specific binding to the ends of the DNA, you will get the amplification of only the desired sequence of DNA. Is it right? Okay. Then another important thing about this enzyme is that it is stable. I have 10 minutes to go. Okay. After that, I will restart the meeting and I will continue till up to 12:30 p.m. Okay. And after my lecture, Buktar madam is going to engage your lecture. Okay. So be prepared for that. And after my lecture, do not go anywhere. Just wait for madam to conduct her lecture. Okay, so this enzyme remains stable even after 50 cycles. Just mind this particular thing. Even after 50 cycles of DNA amplification, this enzyme Taq polymerase will remain stable. About 65% of the activity of the enzyme is still retained. Okay, and that is why we say that this is a very stable enzyme at a high temperature, and that is very useful for carrying out PCR reactions. Okay. Did I tell you about this enzyme? If we carry out few cycles of DNA amplification using the Kleno fragment or DNA Pol1, then most of the activity of the enzyme is lost. And therefore, as Inamul told us, that we have to go on adding the enzyme for every different cycle. Okay. And another important advantage of using Taq polymerase is that longer fragments of DNA can be amplified. Okay, can you see this number 10 kb? 10 kb means 10,000 bases of DNA can be amplified, or that particular DNA can be synthesized using Taq DNA polymerase. But if we compare that with the Kleno fragment, then we have already seen that Kleno fragment can only amplify about 400 base pair. Okay, so there, see this comparison, 400 base pair versus 10,000 base pair. So that is another advantage of using Taq polymerase. Okay, can you see this picture here? This picture is of the hot water spring, okay, which is also called as Mushroom Spring in Yellowstone National Park, USA. And the organism Thermos Aquaticus was actually isolated from this particular site. Okay, and can you see this person standing here? Can you see?
Yes, sir.
Okay, let me make it a little bigger. Yes, so this person standing here is the scientist, Thomas Brock, and he is responsible for isolating this thermophilic organism, which has become very, very useful in molecular biology. Okay. Yes, then we go to another interesting information, and that is different types of thermophilic archaebacteria and hyperthermophilic archaebacteria have been used for isolation of different types of enzymes. Okay, so Taq polymerase is not the only one enzyme which is thermostable. There are several other types of enzymes also. And why these different enzymes are required? Because the major drawback of using Taq DNA polymerase is that it does not have a very good proofreading activity. Okay. For example, we have already seen that the 3' to 5' exonuclease activity, which was responsible for repairing the wrongly incorporated nucleotides. Okay, that is proofreading activity. That proofreading activity of Taq polymerase is not very good, right? And therefore, we need some more different types of enzymes which are more fidelity. We say, okay. Have you heard this term fidelity anytime? This word fidelity?
No, sir.
No. Do you have your mobiles, or can you just go to the Google site and find out the meaning of this term? Instead of just sitting and listening to me, I think we should do some exercise also, isn't it? Are you all doing it?
Faithfulness.
Yes, try it. Just enter this word fidelity in your Google. Okay.
Loyalty.
Yes. What is it?
Loyalty.
Loyalty. Yes. Very good. Loyalty. Anything else? Synonym of this word?
Faithfulness,
I couldn't hear you properly.
according to the book.
Yes, Josh.
Said devotion. Said devotion or
No, I couldn't hear you properly.
Quality of faithfulness or loyalty.
Yes, faithfulness or loyalty. So here actually it means the ability to exactly copy the DNA. Okay, faithfully copy the DNA. Is it right? Okay, so enzyme which is able to exactly copy the template strand, okay, that kind of enzyme is always desirable. Any enzyme which is going to incorporate wrong nucleotides in the DNA sequence are not good for us, okay, because we want to amplify a particular DNA sequence. It may be a gene, and that gene may be used for cloning experiment later, okay. And then if it is a gene for a particular enzyme, and if there is a misincorporated or wrongly incorporated nucleotide, that is, there is a mutation, okay, and therefore that enzyme which is going to be produced from the gene may not be very active. It will not be functioning properly. Okay, so we want the DNA sequences to be amplified, and these DNA sequences should be exactly like the original DNA that we had. Okay, that is called faithfulness of copying the DNA or polymerase activity, right? So dark DNA polymerase has that problem. It is not a high fidelity enzyme. Okay, have you heard this word hi-fi anytime? Hi-fi. You might have heard it many times.
yes, sir.
Hi-fi सुना है ना आपने सबने?
जी.
Yeah. So hi-fi का मतलब क्या है? What is hi-fi?
Sir, upper status maybe.
Huh? What is it? Couldn't hear
Sir,
you.
upper status maybe. Upper status.
Upper status, हाँ, यही मतलब हम लोग निकालते हैं. For us, the meaning is like that. Okay, people belonging to a high status. Okay, we call them as hi-fi people. है ना?
Standard.
But the real meaning, actually, the full form of hi-fi is actually high fidelity. Okay, high fidelity. Right? So hi-fi is actually a short form of high fidelity. Right? So Taq DNA polymerase is not a hi-fi enzyme. That is to say, it is not a high fidelity enzyme because it introduces errors in the DNA sequence. Right? So there are other thermostable enzymes. For example, one organism called Thermos Aquaticus. Can you see this word Brocchianus? This word is taken from the scientist Thomas Brock. Okay, you have already seen his photograph beside the mushroom spring. Okay, so this organism is in his honor. The organism has been named as Thermos Aquaticus, and the enzyme which is taken from this Thermos Aquaticus polymerase enzyme, that is called as TBR. Okay, TBR enzyme. Okay, what is TAQ? Taq polymerase. What is the full? What is this term taken from? TAQ.
Thermophilus aquaticus.
Thermophilus aquaticus.
Thermophilus aquaticus. Okay, so similarly, this TBR is taken from Thermos Aquaticus. Right? Then there is another enzyme which is called T4 DNA polymerase. Okay, T4 polymerase, which is also a good enzyme because it is a high fidelity enzyme, and therefore it will copy the DNA very faithfully, very loyally, and that DNA will be exactly like your original DNA. Right? T4 polymerase. Do you know what is T4?
So it is a kind of phage, I think.
It is a phage, bacteriophage. Very good. Okay, so what is a bacteriophage? It is a virus which infects bacteria, particular types of bacteria. Okay, there is a lot of specificity between the virus and the host, isn't it? So what is the meaning of T4
sir.
polymerase? What is T4 polymerase? Yes, कौन
सर
सा?
सर T4 का मतलब जीनोम बैक्टीरिया के अंदर जो रेप्लिकेट करेगा वो।
क्या वापस बोलो once again.
सर T4 वायरल जीनोम मतलब बैक्टीरिया को बैक्टीरिया के अंदर जो रेप्लिकेट करेगा वो है सर।
Yes, very good. So that is called as the bacteria which are lysogenized with T4 phage. Okay, can you hear what I'm saying? Okay, it is a E. coli
यस सर।
phage. Okay, so E. coli cells which are lysogenized with T4 bacteriophage. Okay, these bacteria are going to produce the enzyme T4 DNA.